So, good morning, everybody. It was planned that Emina gives this talk, right? But unfortunately, Emina fell sick, right? And she's recovering. I prepared the talk, and I can give the talk also because we are closely collaborating with each other, right? And we did do this on the order of 10 years so far. And I want to present something that is from the last, from 2024 and 2020, from results from 2024 and 2025. At the beginning, I really didn't have any idea why I got myself into it with this project. You remember that we worked on PYP for a long time and did super successful, exciting X-Cell experiments with the PYP. And then, of course, the phytochrome would have been something that, I mean, would extend that blue light receptor into the red light regime. So what is this all about now, right? With these phytochromes, and Emina was available, and she had a phytochrome that turns out to be extremely important and extremely interesting. So suppose you do signaling, right? Light signaling or any other signaling, and you do this in a cell, like here it's shown the cell of a bacterium. Wow. Pointer option, laser pointer, I see. The cell of a bacterium. Then typically you have, for example, a membrane-bound receptor that senses a chemical. It's a chemotaxis receptor that where the chemical binds on the extracellular side and the signal is transferred into the intracellular side. And then there is an enzyme. It's an enzyme receptor. And then you see that's little written here, kinase, and it's a typical histidine kinase, right? Typically, this is histidine kinases. And the histidine kinase make then the signaling into the cell, and the signal will be transported by so-called response regulators, which are shown here. Response regulators pick up a phosphate from the histidine kinase, and they move, for example, to pili that are related to the motion of, or to the movement of the bacterium, and also some others which are transcription factors. At the end, they generate the physiology or the metabolic output of the bacterium. Right? But if you have light, for example, I want to say that this is called the two-component system signaling. And the two-component systems, they were invented or they were discovered in 1986 by a Harvard geneticist, Osu Bell. Fred Osu Bell, he's now 80 and is retired, right? But anyway, since then, they are the two-component system signaling pathways. And you can imagine if you want to perceive light. Light is also a stimuli that needs to be leads to cellular responses. You probably don't need a membrane-bound receptor. You can have a receptor that is soluble, so you don't have to deal with membrane proteins, and that also has a histidine kinase that the receptor is light sensitive, has a histidine kinase, and also phosphorylates a response regulator. And we are interested in these red light receptors that sense light from the sun and lead to changes in the morphology and in the physiology of the bacteria. So what kind of bacteria are we looking for? We're looking for Myxobacteria, which are kind of unknown in the United States. They're more known in Germany, for example. There's a big group who works on Myxobacteria, and there are some specialized groups all over the world who work on Myxobacteria. What are these? They're predatory eubacteria. They live in soil, they eat other bacteria. So they must have some chemicals that they secrete to kill the other bacteria. And when they are starved, they want to reproduce and therefore form so-called fruiting bodies. They're multicellular aggregates. You can see them by eye. And they also secrete secondary metabolites with antibiotic properties. Yeah, they need to kill the other bacteria on which they predate, and anti-cancer properties. Turns out that some of these metabolites also can be used against cancer. So these are kind of also biomedically highly relevant bacteria. And the interesting thing is the fruiting body formation is can be controlled by light, right? So for example, if you shine blue light on them, they have photoactive yellow protein, they sense the blue light, make fruiting bodies. If you shine red light on them, there's no fruiting body formation. And if you do far red light, 750 nanometers, they love to have fruiting bodies. So you see that you control with light the fruiting body formation here. And that goes even further. If you pipette the Myxobacteria on a spot on a paper, on a filter paper, and leave them in the dark, they start to grow and they start to migrate and they form fruiting bodies. And they form fruiting bodies in these concentric rings, which means they rest a while, form fruiting bodies, then they start to migrate again for a while, form fruiting bodies. So they must have an internal clock that determines this, the rings and the separation of the rings from each other. So that's in permanent dark. But you can disrupt this inner clock by shining light on it. You shine red light on it, you disrupt this beautiful growth. So they must have a red light receptor in them. And that was all published in a paper. I was not involved, but Emina was involved together with a collaborator at UC Davis, at the University of California in Davis. And it was published here in the biochemical and photobiological sciences last year. So what are these red light receptors? Of course, the candidates are bacterial phytochromes or phytochromes. And the phytochromes, they were discovered in the 50s of the 20th century, right? So famous work on it, they were discovered in plants. And there were lots of people who tried to make use of the phytochrome by shining red light on seedlings and hoped that the seedlings were germinating quicker, right? And therefore, the phytochromes had to do a lot with plant growth, shade avoidance, growth towards light, and other physiological responses. And then no wonder they were also discovered in photosynthetic bacteria because they also have to mature their chloroplasts, or they are basically chloroplasts, right? But they are also found in non-photosynthetic bacteria. They were around the year 2000, right, in the lab of Rick Viestra. They found the phytochrome in non-photosynthetic bacteria. That was quite a surprise, actually, in Deinococcus radiodurans, which has really very little to do with light perception, right? But here you have the Myxobacterial fruiting bodies and the secondary metabolites, which is an underexplored source of natural products. And I want to show you some of these metabolites by going back to here. So you see that filter paper is almost white. And if you go one back, you see this yellow goo that is seeping into the filter paper. And it would be kind of interesting to look at what that is. What are these Myxobacteria secreting? As a function of light exposure, of course. And you see that light has quite an influence on the secretion of these secondary metabolites actually know, at least they secreted in the dark and not in the light. So how do these bacterial phytochromes look like? So this is an AlphaFold model, right, of the full-length bacterial phytochrome. Nobody has a structure of the full-length bacterial phytochrome with the histidine kinase here. There are full-length bacterial phytochromes, but a very limited amount, and they don't have a histidine kinase. They have other little domains sitting here, which sometimes without even knowing what they are doing. And since this is an AlphaFold model, the chromophore, which sits here, is left away, right? So the chromophore that sits here, and the chromophore is a so-called biliverdin. It's a heme derivative, comes from heme. It's a linear or an open chain heme. So you cut the heme open, and then you have this ring A to D configuration of the chromophore. And if you look at the isomerization status, this is Z, this double bond is Z, and this double bond is also Z. So it's an all Z. And then if you look at the rotation about the single bond, there's syn, and this ring points in the other direction. It's an anti configuration. So it's all Z, syn, and anti. So then you can look at some modules here. This thing here that receives the light and transfers the light signal to the histidine kinase. That's called the photosensory core module. It has a roughly 110 kilodalton, right? And this is responsible for the photoreaction. And this is the photoreaction is driven by a cis-trans isomerization of that chromophore called biliverdin, right? So the question is, how is this light then transferred to the histidine kinase from the position where the chromophore is? And how does that light regulate the activity of the enzymatic domain? So far, the people have not crystallized any of these full lengths, but the people have crystallized, and Emina has crystallized the PCM, the photosensory core module, right? And over the years, we solved the structure to a higher and higher and higher resolution. So in the moment, we are at 1.4 angstrom. And you see here, this is the electron density of the chromophore, of the biliverdin chromophore. You see here different domains called pus, and here in green the GAF, and in red the phi domains. So this photosensory core module consists of 3 domains, pus, GAF, and phi, which are found in all phytochromes, even the phytochromes of the plants. And what they also have here, there's an extension of the phytochrome called the phi domain called the sensory tongue that is in contact with the chromophore here. And here the same. And we have 2 subunits, of course, 2 protomers. People call them protomers. I want to call them subunits because it's easier for me. And so the resolution is 1.4 angstrom. This is a record. Nobody has PCMs with higher resolution. I mean, this is already quite good because we can characterize the angles or the configuration of the chromophore to very precise values. So the interesting thing is that, of course, since it's a photoreceptor, the photoreceptor shows a photocycle, like every photoreceptor, like PYP has also nice photocycle, right? So it goes from a PR, why is it PR state, red absorbing state, to a PFR state, the far red absorbing state. So this absorbs at 700 nanometer, that absorbs at 750 nanometer. And you can drive the photocycle by shining light on the PR state that goes over to the PFR state through several intermediates. During the transition from PR to PFR, nobody has seen all these these intermediates. So we are highly interested to see these intermediates. And nobody has seen during a photocycle the appearance of structural changes that are commensurate with the PFR structure. So when you shine light on it here, you'll see first of all that these crosswise helices, right, they widen, open up, and lead to a separation of the phi domains here. And at the same time, the beta sheet here, or the two beta strands, separate and form an alpha helix. So the beta sheet to alpha helix, that is a signature event that shows the PR to PFR transition. And then this may lead to the separation of the phi domains. How do you know that? Well, the people tried to crystallize the PFR structure, and they succeeded. There was a Nature paper by Sebastian Westenhoff in 2014 who reported on this. And then later also in the Viestra group who also showed this. For the Deinococcus radiodurans PCM, but not for our phytochrome, which is from Stigmatella aurantiaca, the Myxobacterium. So, of course, in order to generate a signal, first you have to perceive the signal, transfer the signal, and regulate the enzyme activity, and also regulate the response regulator binding. I told you the response regulator has to bind. It will be phosphorylated by the enzyme, and then it has to dissociate. So these are fundamental events that happen in this photocycle of the enzyme of the enzyme receptor that needs to be addressed. So in order to do this, of course, the protein has to change its shape, and of course that requires methods to look at how shapes can change, right? And then, of course, we can think about this rotation is probably a motor, and then since we have to catalyze a reaction, this protein resembles a chemical reactor that takes a substrate, makes an intermediate, and then finally makes a product. And then also the protein must change shape to transduce the signal, right? It's similar to a mailman, right? It picks up some letter, goes to its car, drives the car to another location, and delivers the letter. And that is all related to shape changes of the protein, right? So this you see already that the static shape is not sufficient to describe the function of that protein. So structural changes have to be investigated. So how do you do this? Of course, you always do this is more or less the same. Prepare a pure state, trigger the reaction, wait a period of time, and measure the structure. And then some people say, oh, structure determination must be faster than the fastest delay. Really, when the reaction happens in 100 picoseconds, for example, in the trans-to-cis isomerization, then you can trigger the reaction with 100 femtosecond laser pulse and determine the structure of the delay of some picoseconds. And then the structure determination must, of course, be done faster than a picosecond, or in the order of a picosecond. But there can be other examples, right? For example, there is a slow photocycle. The photocycle is only complete after 1 second. Then prepare the molecules in the PR state at the beginning of the photocycle, trigger the reaction with CV laser light pulses, only 10 milliseconds, but 10 milliseconds CV laser light exposure. Then freeze the protein after 100 milliseconds, 200 milliseconds, and 300 millisecond delay. So you see that you can freeze it, and then you stop effectively the reaction and preserve the ensemble in a certain state. So then you have plenty of time to determine the structure. Yeah, no need for the fast structure determination is really not necessary if you are on that slow time scale. So I show you some experiments here. We did a lot of experiments over the years. So from the static PR, which you have seen here already, the 1.4 angstrom structure, to the full-length phytochrome, bacterial phytochrome with cryo-electron microscopy. And then at the end, you see the physiological response, which I have shown you already. So how do we start a reaction in phytochromes? Well, this chromophore absorbs somewhere in the red, right? You have this PR spectrum. So in order to start a reaction, to drive the photocycle from PR to PFR, you start the reaction with 640 nanometer light. And then, of course, you do an X-ray probe and probe the structure. And that can be done at a synchrotron, it can be done at an XFEL, dependent how fast you want to probe that structure. So what we usually use are microcrystals, and we do serial femtosecond crystallography at XFELs. But you can also take the same microcrystals and go to the ESRF and make a synchrotron serial crystallography, time-resolved synchrotron serial crystallography with the same nice crystals. They diffract to roughly 2 angstrom with the single pulses, and that's enough for our to show structural changes. And we did actually use these crystals at a number of XFELs, right, at SACLA, LCLS, and European XFEL, and also we used it at the ESRF with several methods to expose the crystals to X-ray radiation, right? So, for example, at the ESF, we use a fixed target, a chip. At the European XFEL, we do liquid injection, and at the other XFELs, we use the Weierstahl toothpaste injector, which injects a viscous matrix. And this is the viscous matrix which we use, the super loop, a nuclear grade grease. So we embed the crystals into the super loop by folding them into the loop. And that's not, we cannot use couple syringes as other people use for LCP for the lipid-based matrices, because the super loop does not allow this. The crystals immediately decompose, they get crushed, there's too much shear motion. So we have to do this by hand. We take a highly concentrated microcrystal suspension and pipette it on a little amount of superloop and use a spatula to mix them until there is a more or less uniform or homogeneous suspension of the crystals and the superloop. Then we fill the superloop crystal suspension into the Weierstahl injector and extrude the grease through, which is shown here on the right side. You see the grease is extruded from that injector, and that works in vacuum, and that also works at ambient pressure, for example, in a helium chamber. So, but you have seen before, we need to have red light to trigger the reaction, and we shoot the red light, of course, onto the jet. And to our surprise, nobody ever investigated the jet reaction to ultra-fast femtosecond X-ray laser pulses. So we had to do it. We had to do it to see how the naked jet actually reacts to a high-intensity femtosecond laser pulses, optical laser pulses. And I show you that. The result here. So this is our geometry, right? So we use the 150 micrometer jet, and we focused the laser to 330 micrometer, and looked how much of that went through the jet by putting a power meter behind the jet, very shortly behind the jet, because we had to make sure that even in the most unrealistic cases, we are covered. In the most unrealistic cases, you see these flanking rays, they refract right onto the optical axis because the index of refraction is so large then that this angle can be negligible. So then the deflection angle here is capped at 2 beta. So we had to come with a power meter as close as possible to capture the 2 beta deflection, maximum 2 beta deflection angle that occurs if in the most extreme and unrealistic cases. But if you don't use pulsed optical laser pulses, but we use CV laser, you can see that here. So that was done by Shavda-Slavov. It's a kind of an interesting experiment. If you use no viscous jet, you get this 90.91, right? What is it, milliwatts? And if you insert the viscous jet, you get 0.82 milliwatts. So roughly 90% of that CV laser light is transmitted. But you see already that this forms this cylindrical lens, right? So the jet forms the cylindrical lens that made us be cautious when we measure really high intensity pulsed laser pulses, because all sorts of things can happen. And we don't know how much is reflected. We have no idea. Here is more normal incidence. Here is more grazing incidence. So the best thing is just to measure it and see what comes up when we do our shine now high intensity femtosecond optical laser pulses onto the jet. And we did that at SACLA. They provided these, they could focus the optical laser to this diameter and provided this jet. And I show you the results. So here we have the nominal dialed in energy, and here we have the measured energy. And this is without grease. So when we dial in 40 or 60 microjoules, right? So we measure 60 microjoules. No big deal. And one can also calculate what the affluence or the energy per unit area is within full width half maximum, which is shown here in brackets, right? And then when we pull in the jet, the naked jet without protein, you see how these kind of transmission problems appear, right? So here from 28 millijoules to 6 millijoules. So if you want to use 6 millijoules for your crystals, you have to dial in 28 millijoules. That's a huge difference. If you want to use 4 millijoules for your crystals, you have to do 14 millijoules per millimeter square. So why do you want actually these high values? If I go back, you see here, we don't shoot in the absorption maximum. We shoot substantially in a substantially smaller absorption than the absorption maximum. In order to do this, we need, of course, more power, right? In order to start the reaction. And when we shoot in the absorption maximum, okay, yeah. But if you are going away from the absorption maximum, you need more power. We did this always at BioCast with PYP, with the idea just to increase the penetration depth, but that is basically the same principle. So if you want to have more power, you run, of course, in these problems, right? If you want to have 2.5 millijoules per millimeter square, you need 7 millijoules, and so on and so on. So, but recently, you remember there was a large criticism of these large laser powers by Schlichting and also Dwayne Miller, that they claimed, when we vary the laser power, we see a change in the relaxation of hemoglobin when you switch photolyze away the CO at high laser powers. The relaxations are much faster than at low laser powers, which is also shown here. This is the low laser power relaxation of residue 93. And that's the high laser power relaxation of residue 93. And the same is true for somehow not so much for the residue 64, right? So there is something that the laser power has done, they at least claim. So in order to do this, first of all, they haven't taken this thin layer, the interaction with a thin layer of matter into account as with the grease. And they didn't really specify extent of reaction initiation. And also they dismissed, at least to my understanding, the negative extrapolated structure factors. So what does that mean, right? So in order to quantify displacements, you need extrapolated maps. In order to calculate an extrapolated map, you add difference amplitudes to the dark state structure factor. And from that, you can calculate this extrapolated map. And if you have NC, if NC is not correct, too small, then your relaxations or your displacements are wrong. For this, you need the extent of, and since NC is related to population transfer or to occupancy, you have to better determine the occupancy rate or the extent of reaction initiation. And it appears it was not reported how they did it for their myoglobin results. So here's this kink method. So you add the delta to the dark state structure factor, and then after a certain point, you get false negatives in the extrapolated map. And the false negatives increase dramatically after this characteristic N, which then determines your occupancy in principle. So if NC is poorly determined, the displacements are also incorrect. And I believe that this is part of the problem that they had. But the other problem was even more subtle. They claimed that they have rejected the negative extrapolated amplitudes. And what is this? When you calculate extrapolated structure factors, you take the dark state structure factor and add the delta. Here, for example, the delta is positive. You add them twice. So you get this blue extrapolated structure factor. You cannot, but the delta F could be negative. You have to subtract them and you get a much smaller extrapolated structure factor. But what happens if the dark state structure factor is small? Has a small amplitude, then of course the extrapolated structure factor points into the opposite direction. That means there are no negative amplitudes. There are amplitudes of a structure factor that point in the opposite direction than the dark state structure factor. And in a refinement program, you cannot dismiss them. These are important cases. They generate contrast. You simply have to think in terms of Babinets principle. So is that really true that you can find laser power differences in Barnes and et al., given that they neglected all these 3 points which I have shown you? But then in addition, Emina went to John Kennis, and John Kennis did the spectroscopic measurements on that phytochrome. And here is the result basically in a nutshell. When you excite with femtosecond laser pulses, you don't only populate S1, but you also populate higher states, SN here, right? But fortunately, John was measuring the decay of SN to S1. This was 300 femtoseconds, right? And he also measured the time constant for ionization was 4 picoseconds. So given these time constants, very little ionization happens. Mostly S1 is populated. And so you see that the S1 is populated through multiple channels, not only by excitation from S0, from the ground state, but also by the rapid decay of higher energy levels. So the guidelines for laser illumination, which were highly publicized by Greenbein and others, and especially by Ilme Schlichting, may not apply to our system. And they might not apply to other systems too. So you need to be very careful when you look at these guidelines that are given by the Schlichting group so far. For our system, it doesn't seem to hold. For our system, we have completely different conditions. We have to look at the interaction with the grease, and we have to also look at the energy, the energy diagram, which is shown here. So I show you the 3 picoseconds, 100 picoseconds results. So these are beautiful difference electron densities. Here in red, there are the negatives, and here in green, there are the positives. And you'll see that they're so easy to interpret, at least here. You see that ring D lifts out of the plane spanned by ring B and C. And this is also true for subunit B, but the angles are different. But then I want to show you also here the pyro water. Pyro water is the water that is bound in the ground state to the dark state to the biliverdin. It is not going away. It stays here. At subunit B, it only moves from here to there. So it also stays. Basically, it's not photo-ejected. Then at 100 picoseconds, subunit B is also, again, easy to interpret. You see that here in green, this is the positive difference electron density, which is nicely interpreted by this orchid structure here. And also the pyro water still stays here. It is not gone, right? Whereas in the subunit A, there is something else going on. There is some additional difference electron density here that we can maybe interpret as a rotation of the ring D from Z to E. So here we see a mixture of Z and E configuration. And part of that pyro water is already on the way out of the chromophore binding pocket. At 33 milliseconds or 5 nanoseconds and 33 milliseconds, the pyro water is finally gone. And we did this at the LCLS in 2022. We got beam time, and Tech was the leading author on this paper that reports on this. So you see a rotation about the single bond that lifts the chromophore, the ring D out of the plane. And then you see a rotation about the double bond that might get us to an E configuration of the ring D. So we see already the onset of the Z to E isomerization at rather at 100 picoseconds in subunit A. So we have an overview for our X-ray sources. We get very good resolution. We get very early intermediates. We get strong chromophore configurational changes and conformational changes. We get small protein structural changes at fast times and more substantial at medium and longer timescales. But what we don't see is this alpha helix formation of the sensory tongue. It stays always beta sheet, and we never see the separation of the phi domains. So the PFR structure of Myxobacterial phytochromes remains unknown, right? And we will never manage to crystallize the full-length intact bacterial phytochrome. So this is then the more in a more schematic overview. Here we have experiments at the LCLS published in Communication Chemistry. It just came out today. Then on the nanosecond to millisecond time scale, where you see that the pyro water is gone and the ring D is in the E configuration, and then there is a block. We cannot go further, at least not with crystallography, and we never were able to crystallize this full-length phytochrome with histidine kinase activity. So that's speculation. It's an AlphaFold model. So what is this missing? I mean, we want to see this, and there's not a full-length structure available. So in order to get the full-length structure, we just try to crystallize it, and you see beautiful crystals when we have when we send the full-length when we try to crystallize the full-length phytochrome. So we expressed it, we purified it, we crystallized it, got it to the ESRF. And then try to solve the structure using AlphaFold model. Of course, it didn't work. The unit cells were way too small, so we must have crystallized the fragment. That is a little bit disappointing, but I show you what comes out here. So rather than to have a Pascaf construct, the fragment was a Pascaf construct as a Pascaf dimer. And we crystallized the Pascal dimer of that phytochrome previously, and you see these are the two subunits, and they have a 6-helix bundle as a domain interface, at a dimer interface. So when we look at that Pascal dimer that is in these crystals, then this dimer interface is different, right? You see that dimer interface is completely different from that dimer interface. It's also a dimer, but the dimer interface is different. It's not the 6-helix bundle. And these 2 helices, they would belong to the 6-helix bundle. Oh, what about crystal contacts? Okay, let's go to crystal contacts. Here, this 2-helix bundle, this is another 2-helix bundle. 1 helix is completely missing. Completely missing. There's no electron density in there. So there must be either been fallen off or so disordered that we cannot see it. And also the 6-helix bundle, which were shown here, are nicely parallel. Here they're perpendicular to each other, the helices, right? So you see a complete different structure, complete different helix dimer interface that we usually have when we have a Pascal crystal from the Pascal construct. So that's already one peculiar thing. But the more funny thing is that actually happens here is that the chromophore is not anymore in the all-Z syn-syn anti-configuration, but suddenly this ring flips to the syn-syn-syn, so the syn configuration. And that means that you cannot close the heme anymore, cannot make a heme out of it anymore. So you have to have you have to stack A and D. So they stack like on a spiral staircase with a pitch of 3.5 angstrom. And you see what large structure changes are required here. But we also see that the histidine 275 suddenly makes a hydrogen bond with the C ring. And that is also observed in the PFR structure. So some of that characteristics of that chromophore somehow seems to be in PFR. But most of this is a decayed construct with an interesting chromophore configuration. And we published this also in structural dynamics. So, but then how to come closer with our quest to look at the signal transduction in Myxobacteria because we need the full-length protein in order to bind the response regulator. First of all, we have to show that the full-length phytochrome is a histidine kinase. And histidine kinases, they phosphorylate. So and they autophosphorylate. In order to autophosphorylate, in order to show this, we need a radioactive assay. There are also other assays, but the radioactive assay is the most sensitive one. And here are the results of the most sensitive one. Here is the spectrum of the full length in PR. Here you see also the autophosphorylation in PR with a radioactive phosphate. Here in red is the spectrum of PFR, and here is the autophosphorylation of PFR. So obviously PFR is not so active as PR, and that was sort of expected from a histidine kinase, from a photoreceptor histidine kinase. It's more active in PR than in PFR. By the way, why I show these two spectra, the photosensory core module spectrum is identical to the full-length spectrum. So the histidine kinase is spectrally completely decoupled from the photosensory core module. So it doesn't matter whether the histidine kinase is there, the spectra are the same. Anyway, so in order to get the full-length structure, we went to electron microscopy, right? Where we asked the Simmons Electron Microscopy Center whether they can help us. Peter Schwander fortunately had some connection, so he could ask Bridget Garager at that time, right? And she was also kind of excited because we can do a time-resolved experiment with this, right? So in order to move forward, we sent them the full length, and we also sent them the PCM. Both of them, we sent them to them. We sent it to them. So they had a small particle and a big particle, and they could measure this. And these are some details of the electron microscopy, which I don't want to go into detail. At the end of the day, we had to make the data processing possible. So they sent us the movies, because nowadays in cryo-EM you make movies, and we use the well-established data processing algorithm CryoSpark, and that provides a user-friendly pathway. And the biggest problem actually was to install it on our compute clusters in order to figure this out. And Peter Schwander and Teg Muller figured this out, and how to run CryoSpark in an effective way on Peter Schwander's compute clusters. Now we actually run it on the UWM compute cluster, but at that time they didn't have one, so we were allowed to. Peter Schwander allowed us to use his cluster. So Tech figured this out and figured out how to process the data, and I show you some details here. So you got these movies, and at the end from the movies you get 1.5 million particles. That looks quite normal, right? So you do 2D classification, and you already see the phytochrome here. What a beautiful 2D class. You see the PCM, and you also see the histidine kinase up here. So we had 2D classes for which we could do an ab initio reconstruction, and one of these classes looks fantastic. So then we did some refinement. After you have this kind of reconstruction, you can go back to the images and ask and refine the orientation of the images, given this as a template. And you get then near atomic resolution. And you can also estimate the resolution by the Fourier shell coefficient, right? So that's an important thing. And you get for each and every power portion of that protein, you get a local resolution, which is shown here from 3 angstrom in the PCM to 6 to 9 angstrom up there in the histidine kinase. So that means that you can interpret the PCM part here with an atomic model. You see side chains and backbone, everything, whereas here you can probably only interpret with some model that you have, and push this and try to refine it into the lobes here of the histidine kinase. So the results are shown here. So we have full-length PR. Full-length PR, you see that the histidine kinase is not that well resolved. The PCM can be interpreted with an atomic model. Both sides, both protomers are in the PR structure, as you see here by this beta sheet of the sensory tongue, right? So, and you can also try to overlay our crystal structure, and you see here in yellow, there is the overlay of the crystal structure. Fits perfectly. But then we could also go to PFR when you shine 660 nanometer light on it. And then what happens here, the 2 strands here separate. You see the separation of these 2 strands, they're very much separated. And on this side too. So you can distinguish PFR simply by looking at the separation of strands. And you see the alpha helix here. Wow, this is the first time that anybody saw a PFR structure almost with a histidine kinase. It's fantastic. But to our biggest surprise, there are, when we don't do nothing to the protein, it's just ambient light exposure, then we found a heterodimer. Here you see that they are really nice together, the strands, and here they're far apart. And this is an alpha helix. So you have the PR and the PFR structure at the same time. And what you also see here, that here, this is PFR, because you see here the alpha helix. And here's a kink in the PR conformation of the opposite subunit. This subunit is still in PR, but the kink appears here. Here is no kink. Here is the kink. So there must be a signal transaction from PFR alpha helix to making this kink in the linker of the linker that connects to the histidine kinase. And here we were able to, the resolution was good enough to put in a tentative model for the histidine kinase, which fits kind of nicely. And it shows that the AlphaFold model was actually not quite correct. It missed the orientation of the histidine kinase lobes. They are different from the AlphaFold model. So when we can now subsume this in this kind of schemes. In PR, right? So you have that cross of this long helices very much in the in the phi domain here. So, and then you can also look at the angles that the particular subunits make. So you see that there are several angles that determine a PR structure and distinguish the PR structure from the other structures. You see, for example, that the histidine kinase, which is here, has an angle with the phi domain. So when you find a histidine kinase that is angled relative to the phi domain, and you have this the linker crossing at that position, you have a PR structure. But in this heterodimer, PR-PFR, the heterodimer, you see that these three subunits are almost aligned. They almost have the same angle relative to the pus domain, right? But what is very important is that the crossing of these long helices now happens up here, way up in the linker. And this is a slide by 50 angstrom. And now you can imagine what happens. Well, we are ready to rotate about this pivot point, right? You rotate about this pivot point and by making these two helices parallel. And that means that in PFR, suddenly the histidine kinase domain is rotated by about 70 degrees relative to the one that is previously. So this is the method. This is the mechanism of activation of that chromophore of that phytochrome. Slide and rotate. Slide and rotate. That's what it is. That's what we are looking for. And this is very, very rational because at the end of the day, this phytochrome needs to get rid of the response regulator. And when it rotates like this, the binding for the response regulator changes and the response regulator might go away. Interestingly, we were also able to do this for the PCM. We found a PR-PR homodimer for the PCM. You see the strands are really separated and you find an alpha helix. But this separation of the phi domains, as shown for the Deinococcus phytochrome, did not happen to this extent. It happened because the crossing of these two helices moved apart. So there's a small opening, but not that big an opening like in the Deinococcus ratidurans phytochrome seen before. And it looks more like the Bathy phytochrome determined by Xiaoxing Yang earlier. So you see that the PFR structure of this of the PCM looks like a Bathy phytochrome in the resting state, which is of course also a PFR homodimer. That is the difference to that what was looked at previously. On the other side, we also find a PR-PFR heterodimer in the small PCM construct. And you see how beautiful this works, how nicely here you see the beta sheet, here you see the alpha helix, and in green we have overlaid here the structure of the full-length PR-PFR heterodimer. So this heterodimer was the sensation of the day. Nobody has seen a heterodimer structure before, at least not in a wild-type protein. We didn't do nothing to this protein. And I will show you in another slide what actually has happened previously. Anyway, so then there's a brand new publication on bioRxiv that looks at the heterodimer. Maybe they can find the heterodimer in a spectroscopic assay. So they use this Pseudomonas aeruginosa bacterial phytochrome, which is a Bathy phytochrome. It lives in PFR. That's the resting state. So in order to make a not equilibrium, get the state away from equilibrium, you do a PR-PR homodimer that will then decay to the PFR resting state. So if you look at that, it decays. And in former times, everybody would say, everything goes to a PFR homodimer. But now with the information that you might have an intermediate here, you can also identify this intermediate, the PFR-PR homodimer, heterodimer, in this return reaction of this phytochrome. And that lives roughly half an hour. Our structure, our intermediate, our PR-PFR heterodimer lives more than a day. So the reaction is very, very slow. So that was our luck that we had that the Myxobacterial Phytochrome showed this almost stable PR-PFR heterodimer that we could find in electron microscopy. Fantastic, isn't it? So I want to show you some more phytochrome structures that came out in recent years. And of course, the most important of the phytochromes are the plant phytochromes. So there is now a plant phytochrome structure of phytochrome B that was published in Nature. And you see that there's again Pascal Phi, right? And then something more, and here Pascal Phi. So you see that they're antiparallel organized. Plant phytochrome has an additional path domain and a long linker, so that at the end of the day, the histidine kinase-like domains are also positioned on top of this raft-like antiparallel structure. So, and then they tried to do phytochrome A, and phytochrome A is even more complicated, and it has more path domains. Here there was only one path domain. Here's path 1, path 2. They also showed this antiparallel a scaffold, a raft on which the histidine kinase or the related domain is lying. So on the other side, this was a good resolution, better than 5 angstrom, but if you have really bad resolution, you find an cryo-EM density that looks like this, and it was very difficult to actually interpret it. And so this structure and that structure obviously is very different from each other. It's not really clear whether that is correct. But at the other, in the meantime, also they tried to make a cryo-EM structure of the Deinococcus rhabdodurans full-length bacterial phytochrome, and they couldn't stabilize the histidine kinase. They had to bind the response regulator to it, and they actually fused one to it. And it's heavier, so it might stabilize the protein. And you see that didn't even help at all. So there is no histidine kinase domain visible. So we were really the first to showed it. And then at the same time, around 2024, the group of Viestra again also tried to do it with Pseudomonas syringae bacterial phytochrome. And you see that also the histidine kinase is not really defined here, right? So we were really the first to show this. And this paper actually came out one day after we published our results with the cryo-EM, right? The only heterodimer so far that was available is an extra structure actually of the full-length bacterial phytochrome that has an effector domain with diguanylyl cyclase activity. And that also crystallizes in a PR-PFR heterodimer, but in order to produce it, it was necessary to make huge manipulations or mutations to the linker region that heterodimer is stable. So, but we got one, a wild type heterodimer that was the great and wonderful results that we got at that time. So we can now say something about the mechanism. We find ultra-fast and fast chromophore configurations that can initiate the signal. Then the signal moves to the opposite subunit makes this kink in the linker helix. And then the crossover of a helices moves up by 50 angstrom in the PR-PFR heterodimer. The histidine kinase is ready to rotate, and then a second photon needs to be absorbed to form a kink in the second subunit, which was not affected before. Once this kink is formed, the histidine kinase rotates and you find the PFR homodimer. So the hypothesis is now response regulator binding is affected due to this slide and rotate mechanism. And note, also in the PFR, the histidine kinase activity is not as large as in the PR structure. So then we get the structural overview. Again, here the early events, right? Later events, the parallel water leaves. And here finally we get full relaxations. And here we also have the full-length bacterial phytochrome. So all the speculation is removed, and we could structurally characterize the entire signaling cascade in this phytochrome. That presumably leads to the disruption of the fruiting bodies that were measured by Emina and so nicely. So a little bit the outlook. Of course, we need the response regulator now, right? And we got the response regulator. We bought an overexpression system from GenScript, right? Made some 250 milligrams of the response regulator, sent the crystals again to ESRF and solved the structure using molecular replacement. And I must also say none of the AlphaFold models worked, and also the previous response regulators, because this thing here, this part was forming an C-terminal helix, which would disrupt, which would not fit into the unit cell, right? So it makes crystal overlap, right? How do you say that? Clashes, which would not work. So here this helix cannot be. Here the C-terminal helix is somehow disordered and gives this kind of loop. And it could really be that that loop is involved in the binding of the response regulator to the phytochrome. Why do we think it binds up here? Well, there are other response regulator, other histidine kinases with response regulator binding, and they typically bind to the 4-helix bundle near the end of the histidine kinase lobes. That's why we think it binds there. So at the conclusion, so we have details of the signaling cascade from which we measured at X-FELS and X-ray sources and other X-ray sources, and which we measured at with cryo-EM. So we combined crystallography and cryo-EM with physiological investigations to make sense of that, what we actually really measure in terms of physiological response. And of course, in the future, we were looking for a thymocyte single-parted cryo-EM experiment, and we are on it in the moment. So thanks for your attention. I want to acknowledge certain people, especially Tech Mala, who was instrumental for the cryo-EM experiments and also for the data processing of the the serial crystallography data, and with our group, which is shown here, and also Emina, who initiated all this research. I would never have thought that we come so far, and she's also actively involved in all the processes we are doing with the phytochromes, including interpretation, protein purification, going to synchrotrons and X-FELS. So, and then our powerful collaborator Sebastian and Petra Fromme, and especially Peter, who's has always an eye on data processing, because he has a lot of experience with electron microscopy, and lots of people who really helped with the data collection and data processing at the X-FELS. These are the important people at the Simmons Electron Microscopy Center. And there's even one from Microsoft, because Hai Gang had some beam time at the LCLS and he couldn't take it. And fortunately, the LCLS agreed that we can take it for our phytochrome. And funding comes from the NSF, right? We have previously we were at BioXFEL, but BioXFEL runs out. And now we have a little grant, right? Enzyme dynamics on biological relevant timescales. Anyway, so I want to show you one picture here. This is the control room of the LCLS when we did that experiment in 2022. And there are 3 students from Emina. So this is Louis, Viri, and Isaiah. They're all first-generation students. Their parents are just are working class people. They come from a very moderate background. Louis is now at Harvard. Viri is, I think, at Northeastern University, and Isaiah is University of Chicago, all in PhD programs. And you see what this kind of effort does to students who are interested, talented, and willing to work with us. So I must really say I'm pretty proud that we got so far with that. And at the end, since it's still recorded, I want, for your convenience, there are some of the publications which are important to understand the research that we are doing on the phytochromes. Everything started with this paper in 2018, and we got a lot of more interesting results so far. Thank you again for your attention, and this is the end of my talk.